I'm here presenting on behalf of Capricor Therapeutics. We're a Los Angeles-based biotechnology company developing biological therapeutics for the treatment predominantly of cardiovascular diseases and other serious diseases with unmet medical needs. We're publicly traded on the NASDAQ, so I'll be making some forward-looking statements. Our ticker is CAPR. So a quick look at the pipeline at Capricor. I'm going to focus today on our cell therapy program. Our CDCs are what we call them, cardiosphere-derived cells, what we call CAP1002. It's in 3 current or recently completed clinical trials. The first is Duchenne muscular dystrophy cardiomyopathy. It's a phase 2 study. We recently completed enrollment in that study. We'll be having data out in the early part of next year. We're also using the cells to treat advanced heart failure. We recently announced the results from that trial, which looked very positive in a small number of patients. And we're in a big study treating post-myocardial infarction and post-heart attack, which is recently completed enrollment as well, which we hope to have the data out sometime next year. I'm also going to spend a few minutes, as Renuron spoke of, speaking about our Exosomes technology, which we're moving into the clinic hopefully next year, treating ocular graft-versus-host disease. So what is CAP1002? What is our product? It's an allogeneic cell therapy derived from donor heart tissue. We believe it functions as a local drug delivery system. It has a bunch of main functions, but the 4 that I really want to highlight today are it prevents cardiomyocyte apoptosis or programmed cell death, promotes angiogenesis, the formation of blood vessels, attracts endogenous stem cells, and very importantly, it's been shown to be anti-fibrotic or anti-scarring, which is very important in some of the cardiovascular diseases that we've been targeting to date in the clinic. Has a good record, good safety record, and we've exclusively licensed our cells from Cedars-Sinai Medical Center, Johns Hopkins, and the University of Rome. And I don't think I mentioned it, but Capricor was originally a spin-out of intellectual property out of Johns Hopkins University. We moved to Cedars-Sinai, where our founder is the head of cardiology at Cedars currently. So our proof of concept study was a study published in The Lancet. It was called Caduceus. It was with an autologous cell therapy, the same cells but an autologous cell. It was treating 25 patients post-MI. And what we saw actually was for the first time we saw heart regeneration is therapeutically possible. And you can look at the graph on the left. You'll see actually this is the reduction in scar size after the MI. You see the white bars were the patients that did not receive the cells, and you see the red bars were patients that did. Then on the opposite side of this graph, you actually see the growth in viable cardiac mass. Again, white bars showing the controls, and the red bars showing the patients that were treated. So remarkable results. This led us to get a round of funding, and we were private at the time, $6 million, and actually led us to take the product out from an autologous model and then shift it to an allogeneic. That was our next step. So, it also led to this, which I'm going to talk about for a few moments, which is our relationship with Janssen, the wholly owned subsidiary of J&J, as everyone knows. That attracted their interest, and they came in and we entered into a license and exclusive option for them to take a look at our data after we deliver the results to them sometime next year. I'm going to talk about that trial in a moment. They'll have a first look at that data, and what the additional milestones and payments could total up to $325 million. There's an upfront license fee as well as low double-digit royalties. So this was a game changer for us, a great deal to enter into with Janssen. Another part that we don't talk about a lot is we're actually in active CMC collaboration efforts with the Janssen team. So we're utilizing what they have in their team, and we're working together with them in an effort to get this product ready for pivotal trials or potentially for the market. We received $12.5 million upfront from this deal. So over the next few slides, I'm going to explain our adult cardiovascular indications. And as I mentioned a moment ago, there are 2, really advanced heart failure and post-MI. So the first one I'll mention is the AllStar Trial. We recently completed enrollment in this study. It was a phase 2 randomized, double-blind, placebo-controlled trial. We had 30 centers throughout the US and Canada. We treated 134 subjects, and they were broken into 2 cohorts, the first being 30 to 90 days after the heart attack, and the second being 91 to 365 days after the heart attack. We infused 25 million cells of our CAP1002 product down the infarct-associated coronary artery, and our primary efficacy analysis is based on a 12-month infarct size reduction or scar size reduction. Again, as I mentioned, Janssen will have a first look at the 6-month data on this trial sometime in the later part or in the middle part of next year, and that's when they will make their decision to license in this product. We did a phase 1, of course, in 14 patients, and we saw, of course, safety, and we also saw evidence of scar size reduction and ejection fraction improvement. Here's a snip of that data. Again, small number of patients, but what we saw here was a 15% reduction in the scar size at 1 year and a 4 percentage point improvement in EF also at 1 year. So this obviously led us to the design of the phase 2 study. It took us about 2.5 years to complete enrollment, which we were very excited to announce a few weeks ago. So we're eagerly awaiting the 6-month and the 12-month data from this trial to talk about it. Dynamic, as I mentioned, was our study in advanced heart failure. So the NIH gave us about $3 million a few years ago, and we decided we wanted to take the cells and move them into the advanced heart failure patient population. Heart failure, as everyone knows, is a major, major problem to the medical system. There's over 5 million people that have it. There's a 5-year, 50% mortality rate. It's obviously a major drain on the healthcare system. I think everyone knows that. So we did a small phase 2A. We treated 14 patients. We actually, one big change from the AllStar study, other than the different patient population, was we used triple vessel infusion in this trial. And we actually used 75 million cells of our CDC product. So that was very important for us to test out, up to 75 million, escalating, as you can see. We had 6 and 12-month data on this trial. For lack of time, I'm not going to present that data, but we saw a variety of positive changes. We saw EF improvements. We saw reduced volumes. We saw very good changes, which we were very encouraged about. We recently presented last week at the TCT conference in Washington, D.C. So now I'm going to talk a few minutes about our Duchenne muscular dystrophy program. This is what we're really excited about. Internally, of course, we're very excited about the heart failure and post-MI trials as well, but Duchenne muscular dystrophy, I don't think I'll spend a little time talking about the disease. I think everyone's heard of Sarepta and what they're doing. We believe our therapy, first and foremost, would be used complementary to what Sarepta is doing, trying to correct the skeletal or respiratory problems that are facing the boys. But one takeaway from this slide is that cardiomyopathy is actually the #1 cause of death in the Duchenne patient population. They progressively develop more and more scar on their hearts, much like a heart failure patient, and that is actually the leading cause of death. There's 1 in 3,600 male births in the U.S. This is the incidence rate. Approximately 20,000 boys suffer from this disease. So we went back to the bench and to the lab and did some preclinical work, which I'm going to show you in a moment, and decided that this would be a great targeted indication for our cells. As I mentioned, they've been shown to be anti-fibrotic now in multiple studies, and we felt this would be a great targeted indication to go after. Limited regulatory pathway. We have orphan drug designation by the FDA. So, and again, a very terrible disease. So that's what we're targeting right now. Their hearts become dilated and non-compliant and actually eventually fail over time. And as I mentioned, there's no approved therapies for heart disease associated with Duchenne. There's one approved therapy in the U.S., and that's Sarepta's, which was recently approved. This is just a snip of some of our preclinical data where we use the MDX mouse model. It's the gold standard when going to the FDA when you're looking at Duchenne. And I'll just spend a few moments talking you through the data. The graph on the left was change in EF, and you can see the red bars were the mice that were treated with CAP1002. The yellow bars were the MDX mice treated with vehicle. And you can see we have an EF bump immediately after the infusion of cells. You can see it begin to wane off, and then you actually see where we did a second infusion of our cells, and you see the EF pop back up, and you see the further decline of the EF with the MDX with vehicle. So, of course, that was very reassuring to us as we move forward with the agency. The graph on the right is a really important graph. It looks pretty normal for the regular eye, but it's actually really important to us. So the blue is the control, just regular standard mice. The red is mice treated with our cells, and the yellow, again, mice treated with vehicle. But what you'll see, this is measuring exercise capacity. So on the y-axis, you see actually meters that these little mice are running on there. See regular control mice are maintaining a steady 650 around there. and you see the mice treated with cells at around 400, and then of course you see the control mice or the MDX mice treated with vehicle down in the 200. So we're seeing other effects that the cells are doing, and this full data set is actually available on our website. I'm happy to talk to you, anyone, after this. But we're seeing a lot of things going on in the heart that we're really encouraged about. It led us to design our HOPE Duchenne study. This is treating boys, again, with Duchenne muscular dystrophy cardiomyopathy. It's a randomized phase 1, phase 2 trial. We did it at 3 centers, Cincinnati Children's, one of the big Duchenne centers in the world, Cedars-Sinai, and I believe a center in Philadelphia. But we treated 13 patients with Duchenne. They received our cells. They received triple vessel infusion, so 75 million cells. They received them throughout the heart, and then 12 we actually have in a comparator arm. So we recently completed enrollment. As I said, this data will be out in March of 2017. We're really excited to present it, and worked on that trial really hard. We actually enrolled it in 7 months. So there's a major, major need for these boys to find therapies and to hopefully improve the function of their heart. So lastly, I'm just going to spend a few minutes on CAP2003. That's what we call our exosome program. So I didn't mention it earlier. Our exosomes are derived from our cell therapy program. From CAP1002 is where we get our exosomes, and we believe the exosomes to be the mediators of the beneficial effects of the cells. We believe them to be one of the main mediators, at least. So we're rapidly advancing them through preclinical development right now. We're targeting diseases of inflammation and a variety of other diseases that we're looking at, and Renan mentioned it. Exosomes they're actually the size of a virus, nano-sized vesicles that can reduce inflammation and regenerate damaged tissue, and they've been shown to do that preclinically to date. They've been used in many applications. We believe they can be used in many applications that maybe our cells can't. Of course, cells are much larger than the exosomes, so we're targeting indications to where the size of the exosomes may be something that is complementary to the eventual treatment and therapy that we may be providing. We have an exclusive worldwide license from Cedars-Sinai Medical Center for our exosome program, and our targeted first indication, so our first step out of the cardiovascular diseases that we've been talking about for the last few minutes is actually ocular graft-versus-host disease. So why did we choose ocular graft-versus-host disease? Well, we felt that the eye was a great area to explore the exosomes, a lot having to do with their size. It's a compartment, it's a small compartment, and we figure the exosomes can go in there and do what they do best and stay there and not be washed away so quickly. So ocular graft-versus-host disease is a common complication of bone marrow transplantations, obviously after cancer treatments and whatnot. It's a common debilitating form of chronic graft-versus-host disease. It's an unmet medical need, and what we've heard from some of the clinicians, this almost feels like someone's getting sand thrown in their eyes. It's a really terrible burning sensation, big inflammation. So again, we're targeting it. It's an orphan indication. We estimate the market size to be about 3,400 new cases per year. We're rapidly going through preclinical development, and we're anticipating being in the clinic sometime in the middle to, we'll file in an IND somewhere in the middle part of next year. Quick snapshot of the cash, my favorite slide because the one I can talk about the most. We raised approximately $10 million a few months ago. Brought in some credible institutional investors. Of course, strengthen the balance sheet was the main goal. And another important bullet point to note is the fact that Capricor has brought in over $30 million in competitive grant funding. So the U.S. Department of Defense actually gave us a $2.5 million grant to fund some of our exosome scale-up work, and the NIH has given us over $10 million to date, and we're also well-funded by CIRM. So a lot of our clinical work, phase 1, and actually 2 of our phase 2 programs have been funded through grant sources, whether it be the NIH, CIRM, and now the Department of Defense. So we've done a really good job of leveraging that non-dilutive capital and then going out and raising capital when we need to. That's our cash position as of June 30. We'll be reporting our quarter end next week, so hopefully I'll update this slide at that time. And what can you look forward to from Capricor? Well, from CAP1002, our cell therapy in Duchenne, you can look forward to the 6-month data on that patient population, probably March of 2017. So we're a short time away from presenting that data. With CAP1002 in adult heart disease, we're looking forward to delivering the data to Janssen, where they will have an ability to take their license option on the cell program. That'll be sometime in mid-2017. As I mentioned, we just completed enrollment in that study. And then with Exosomes, we hope to submit an IND application for ocular graft-versus-host disease sometime in the middle part of next year. And just a quick snapshot of our board of directors, just give them some recognition if you didn't know any of them. Great board of directors, and Linda Marban, our CEO, who's not here today, and Frank Litvak, our executive chairman, back in Los Angeles. Thank you for your time. I appreciate it.